We can now welcome our last speaker, Dr. Snigdha Mukhopadhyay from the Sorbonne Paris North University in France. He will talk about a trade-off between food quality and transportability during high temperature foraging in ants.
Hello. Hi.
Hello.
Hello.
So you can now share your screen.
Yeah, I'm sharing my screen. Okay. Is it visible full on full screen?
It's perfect, yes.
Okay. Should I start now? Hello.
Yes, you can start.
Oh, okay. So hi, I am Snigdha, a postdoctoral fellow from Sorbonne University, Paris North. So today I will be presenting a part of my postdoctoral work, trade-off between food quality and transportability during high temperature foraging in the thermophilic and Aphaenogaster. So foraging, we all know it is a process of searching for food and acquiring the food, which provide us energy for our survival. And this process is energetically costly, as well as this process possesses potential risk from the surrounding environment, like harsh temperature, rain, then pressure from the predators, as well as the competitors. So animals are known to optimize their foraging process in such a way they adopt certain foraging strategy to enhance the net gain energy or maximize the net gain energy at a cost of lower energy expenditure. And they do it or they adapt certain strategies like spatial or temporal separation of foraging niche to reduce the pressure from the competitors. Like some species, they prefer to forage at very early hour of the dawn or dusk, when most of the species, they do not forage. So that actually reduce their competition pressure. Then some species, they control the recruitment of the foragers to the food source. Like we all know, in honeybee, they do dancing. There are two types of dance, like round dance and waggle dance. It depends on the food quality as well as the location or distance of the food source from the nest. And many species, they are known to minimize their foraging path to optimize their whole foraging process. In ant, three types of foraging recruitments have been studied till date. First one is tandem running, where one individual has the information of the foraging sites and she recruits her nestmate to the foraging sites one at a time, and this recruited nestmate become the recruiter and recruit other nestmate over the time. Second type of recruitment is chemical mass recruitment, which is very common. We have seen in our day-to-day life that a trail of ants, thousands and thousands of ants are coming to a certain food source. Even if you drop some honey, you can find that lots of ants are approaching to that particular honey drops. So here, some individuals, they found the food source and then they lay the trail from the food source to the nest. And that trail, the nestmate, they follow that trail and reach the food source. And when they are going back to the nest, they reinforce the the pheromone trail by again lay some more pheromone. Third type of recruitment is group recruitment, where individual has information of the foraging or the food source, and one individual can lead maximum of 10 to 15 fellow nestmate to the food source. This is not as havoc as the chemical mass recruitment, but here some little amount of trail pheromone used. So the model system I am using, it's a very interesting model system, which called Aphaenogaster senilis, also known as Mediterranean gypsy ants. This ant belong to subfamily Myrmicini, and they share their habitat with several other species like Lesusnezar, then Cataglyphis, Temnothorax, Tapinoma, many more. So what they do that to avoid their the pressure of the competitor over their food resources, they prefer to forage at higher temperature. That's why they call thermophilic ants. And they mostly forage around 45 to 50 degrees centigrades. And these ants show a very interesting behavior, foraging behavior. They can switch between individual foraging to group recruitment based on the food quality, quantity, as well as food transportability. So our objective of this study is to check that do these ants optimize their foraging activity at higher temperature? And also we wanted to know that do these ants balance the trade-off between food quality and food transportability at high temperature? So to answer this question, we conducted sets of lab-based experiment with 8 subcolonies of Aphaenogaster senilis, which consist of 100 adult ants along with 50 larvae. And we allow them to forage in a 28 centimeter by 56 centimeter foraging arena at 25 degree and 45 degree centigrades. To maintain the base temperature of the arena base, we placed the foraging arena on a heating plate, and we provided them 2 types of food. One is easily transportable sesame seeds, which is as the quality of the food is fat and fiber rich, whereas we provided them another type of food which is protein rich but bigger in size, heavyweight, and that needed cooperative transportation. At least 2 to 3 individual is needed to transport it that back to the nest. And we starved the colony for 7 days. Here what we have done that within the 2 condition of the experimental temperature, we gave them a 90-minute of resting period. So what we have found that during both the temperature foraging event at 25 degrees centigrade as well as at 45 degrees centigrade, both types of foods were transported back to the nest. And then we check that the discovery of the first food plate, and we have found that at high temperature, the first food plate discovery time was significantly lower. They discovered it faster. Then we wanted to check that whether the food type has any influence or the placement of the food plate has any influence on the discovery time or not. We have found that the food type did not influence at all in the discovery of the first food plate. But every time, at least 75% of the time, we have found that whatever the food placed at the right side was discovered first. So we can say that high temperature and placement of the food plate influence the discovery time of the food plate, and at high temperature, the food plate discovery time was faster. Then we check the number of the foragers, and here we have found that the number of foragers was significantly lower at high temperature foraging. Then another parameter we have analyzed, that is mass delivery rate of cricket. Here I would like to explain the particular parameter a little bit, because the cricket was transported back to the nest cooperatively. That means more than two foragers needed to transport the cricket back to the nest. So this particular parameter actually dealing or actually explaining that the mass of cricket transported per minute by per individual transporter. And here we have found that at high temperature, the MDR or mass delivery rate of cricket was significantly higher. Then we check the seed transportation rate because the seeds were transported by individual foragers. So we just check that the weight of the milligram of the seeds transported back to the nest per minute. And here also we have found that at high temperature, the seed transportation rate also significantly higher. Okay. Then we wanted to check that while both the foods were transported simultaneously, how do they balance at, the transportation dynamics at high temperature? So here we then microanalyze the seed transportation rate with respect to the cricket transportation. And we divided the seed transportation phase into 3 phases. One is before transportation of the cricket, during the cricket transportation, and after the cricket transportation. And we have found that at 25 degrees centigrade, that the transportation of the seed transportation rate was consistent before, during, and after cricket transportation. But in case of high temperature foraging, or that is the 45 degrees centigrade, we have found that before and during phases, the seed transportation rate was significantly lower. Only after all the crickets were transported back to the nest at high temperature, then only they increased the seed transportation rate significantly higher. So, throughout this study, we have found that both the foods were transported, or both the foods were collected back to the nest. The crickets were transported by cooperative transportation. The seeds were transported by individual forager. But in the lab condition, we did not found any active group recruitment. Then we can say that these ants optimize their foraging activity at high temperature by lowering the number of foragers. Here they are regulating the foraging activity and optimize the process, as well as the increasing the mass delivery rate of the cricket and the transportation rate of the seeds. And we can say that they can trade off between the food quality and food transportability by showing an inclined preference towards the heavyweight protein-rich food, that is cricket, and that is slightly difficult to transport it back to the nest. And these are only increased or only focused on their seed transportation after they are transporting all the crickets back to the nest. So, as a conclusion, we can say that these ants optimized their foraging activity to choose the preferred protein-rich foods, which is cricket, over its difficulty in transportation at high temperature. So, to conduct this study, my team leader, Professor Patricia Detor, she helped me a lot, and as well as our lab manager, Paul Devin, he also helped me a lot. And then I would like to thank the laboratory where I am working, Laboratory the Ethology Experimental at Compre, and Université Sorbonne Paris Nord, and my grant agency, fellowship grant agency, Fission Foundation, and the organizer of Animal Behavior Life. And last but not the least, to all the audience for your precious time. I am ready to take any question. Thank you.
Thank you for this great talk. Like, it's a nice experiment. So we already have questions. From David, in high temperatures, do more foragers go to each cricket so that they can move it faster, or even if fewer foragers are out?
Sorry to interrupt you. Should I stop the sharing?
No, you can leave it like this.
Okay, okay.
And just
Okay, yeah. Can you repeat your...
Yeah, I can repeat.
Yeah.
In high temperature, do
Yes.
more foragers go to each cricket so that they can move it faster?
Yes, we also check the number of forager for each cricket's transportation, which is not significantly higher, but we have found a little bit number of, it is not significantly higher. So I did not present that graph, but Yeah, sometime we have found that in case of the 25 degrees centigrades, 4 to 5 foragers, they are transporting the cricket, each cricket, whereas in case of high temperature, it is like around 5 to 6 individual. And these crickets are really big. It's like 300, around 300 mg. So it is really big to transport. Yes,
Okay.
but it is not significantly different.
We have another question from AB.
Okay.
Great talk. Oops, sorry. Great talk. I'm not sure I understood why there is a right bias in the discovery of the food plate. Are they responding to some external cues?
Here, I cannot say they are using external cues because we block the surroundings with I mean the foraging arena has a little bit of elevation. It has an wall, and this is surrounded. This is actually covered with completely white papers. So I don't think, and on the top of the arena also we put on the light, which is uniformly lighted the arena. So I don't think they are getting any external cues, but and I haven't tested their side biasness in any other experiment. It's a good question, so we can check and we can perform small experiments on their side biasness. But as far as I know, they are not side biased. That is that I know, but yes, like we can do that surely. But I know that like it is already done or tested that they are not side biased.
Okay. We have another question about the temperature. How did you choose these two temperatures? Do you know what temperature? Oops, sorry, it's moving. Do you know what temperature range this species encounters in its distribution range? And what do you only test two temperatures?
Sorry, can you? I could not understand.
It's about the temperature you choose. So why did you choose these two temperatures? And is it the normal temperature for this species?
So these species are diurnal species. So 25-degree temperature we have used because in the lab condition where we are rearing the colonies, because these colonies we collected from another place. So we brought them back to the lab and we rearing them at 25-degree temperature. So that is the control temperature. We are rearing it. So that's why we use the 25-degree temperature as control temperature. And 45-degree, yes, their foraging range is 45 to 50 degrees centigrades. So that's why I have used it, this temperature.
That's perfect.
And I guess, for 45, 46 is their optimal temperature. They do the higher activity, and this is published data. So that's why we have chosen 45 degrees centigrades.
That's perfect. And we have another question here from Catherine. Hello, and thank you for the talk. I was wondering if the short to discover the first plate at 45 degrees is due to an increase in walking speed.
Sorry, increasing?
I was wondering if the short latency to discover the first food plate at 45 degrees is due to a higher walking speed.
Okay, yes, that means every time 45 degree came later before the 25 degree. Yeah, I can understand. So, yeah, because what I have done here is in the nature also they are diurnal species, so they prefer to forage during the daytime, and that's why in the early hour of their foraging it is 25 degree. So they rarely do foraging at 25 degree, and they mostly do it at higher temperature. That's why I kept it in this way. But in another sets of experiment we have done where we randomly allow them to forage at 45. For some subcolonies, we allow them to forage at 45 degree first and 25 degree later. For some colonies, we do the vice versa, and we found same type of result. I mean, the same pattern of result. Maybe, I mean, yeah, maybe their foraging activity is higher at the temperature, so maybe they are just showing faster latency for the transportation or for the discovery.
And I have one last question,
Yeah, sure.
from just for me, a quick
Yeah,
one.
I'm happy to answer any question.
Why would they increase this food transportation at higher temperature? Are they preparing for a dry season or something? Like, either an ecological reason for this?
Yeah, it's a very good question. Yes, these ants, they went, I mean, they go to hibernation during the
Okay.
winter.
Dry?
I mean,
Oh.
yeah, at the end of this month, I will put all the colonies into hibernation. So yes, they do. I mean, they do storage for the
That's why. Okay. Thank you
winter.
very much.
Thank you.
It's the end of the question period. You can still go on Discord, because I
Yeah,
think we
yeah,
will
I
have
will.
much more questions. Thank you for your
Okay.
talk. You can leave the station now.
Okay. Bye.
Bye.
Bye.